Good evening, everybody. Good morning, good afternoon, wherever you are. I'd like to welcome you all to this very special webinar brought to you by the International Academy for Clinical Hematology. And this has become now a tradition to have a post-Congress special webinar roundtable discussion, post-ASH, post-ASCO, post-EHA. We'll have post IMW, I think, later during the year. But today, this is about post EHA and post ASCO 2022 with a special focus on multiple myeloma. And I'm very glad to have with me today some top experts in this field. Professor Meral Beksak from Turkey, Professor Mariví Mateos from Spain, Professor Herman Einzeli from Germany, Professor Rakesh Popat from the UK, Professor Salomon Manier and Thierry Facon from France. And actually, we still have also another couple of colleagues who will join, but who are a bit late. But in order to not to lose the momentum, we'll start the discussion. And as you probably know, and you may be familiar with the IACH activities, I'm sure, actually, we're not going to present any slide, because you've seen all the slides several times. There's a lot of repetition, and you can get them, I think, from the speakers. You can get them on Google. You can get them on Twitter, a screenshot, and you get everything. Actually, the spirit of this activity is really to have a critical analysis of all of these data, not always, the official take-home message saying promising efficacy and manageable toxicity. That's great, but that is not what we would like to achieve. And this is really, I was just mentioning that freedom is a driving force behind this activity. So please, guys, don't hesitate to express your opinions very strongly about how do you feel the data was and where it should take us, and maybe how to interpret this data. And in order to structure this discussion, because otherwise it will be a big mess, I thought that we can have three major chapters. One chapter dedicated to first-line treatment and especially transplant, because we had a presidential abstract at ASCO. This is namely the determination trial. Second chapter, which is slightly linked, I would say, to the first line, is the treatment of high-risk multiple myeloma, because we also had some nice communications about high-risk multiple myeloma. And then the third big chapter would be about immune therapy and the amazing number of bispecific antibodies and data that were presented. And we'll try to put this into perspective with the CAR T cells, although I haven't seen so many CAR T cells new data, I would say, but still, I think all of this is really important. So, okay, let's start now the discussion, and the starting point is going to be the determination data. And of course, please feel free to, I do apologize because I would distribute the microphone based sometimes on how people are on my screen, which doesn't follow any special logic. I've never understood, the logic of Zoom, how people are distributed. I think they are by order of arrival or whatever. Anyway, so I'll start. I can see Professor Beksak first on my screen. The determination trial, Meral, and this is the famous phase 3 randomized trial that was presented by Paul Richardson, comparing a non-transplant arm mainly based on VRD followed by LEN maintenance versus VRD transplant and LEN maintenance. And the key information is that maintenance was given until progression. So, and there is a significant difference in PFS, but similar overall survival. And here would like to hear the opinion of the panel about their interpretation of these data, because it can be the glass is half full or the glass is half empty, depending whether we consider PFS or OS, and where the field of transplant is going. Meral.
Well, thank you, Mohammed. Thank you for the invitation and being here among the experts. I think below the age of 65, this trial is the twin study for the IFM 2009. Maybe not exactly one egg twins, but they differ based on the maintenance phase, because in the US, I think based on their financial status, they prefer to continue maintenance until progression, whereas in France, it was limited to 1 year. And so the results are very similar in terms of overall survival, but I think everybody, and you here from the IFM experts would like maybe to take different points of discussion, but the progression-free survival, the differences are different, and everybody is explaining based on the maintenance duration. In one study, in the IFM study, the advantage is 30%, whereas in the termination study, it looks like a little better. I think in the future we will hear more about the subgroups and the role of transplant about the age of 65. I think that is the one which really needs to be discussed further. Below the age of 65, I don't see any question. It is valid.
So your interpretation is that transplant is here to stay?
Sure.
Okay, Marivi.
Yeah, overall, transplant is here for staying because of the benefit in progression-free survival, but I would add two important things. Point number one, there is not any benefit in overall survival. And point number two, when the measurable residual disease is incorporated, my interpretation is, well, patient preferences can be taken into consideration because of the lack of benefit in overall survival. And the second important point in the future, maybe the risk status together with the measurable residual disease evaluation can help us to identify patients in which maybe we can say them, you don't need a transplant as part of the first line of therapy.
But then, Marivi, I always hear this comparison between early versus late transplant. But when you look to the French data, well, this dogma is probably right, because roughly 70% of the patients are able to get a late transplant. But actually in the US practice, I don't think it's relevant, because less than 30% actually manage to get the second transplant. So,
but they receive the optimal salvage therapies. And maybe this is the effect, well, or this is the fact that makes at the end that we don't see any difference in overall survival.
Okay, but then you prefer to focus on the PFS benefit and continue to offer transplant. Am I correct?
Today, I would say yes, but I would like to wait to know the data from the MIDAS study conducted in the French group of the Master 2, in which the question about transplant, yes or not, adapted to the minimal residual disease is going to be evaluated. And this is the reason why today I continue offering transplant to my patients. But if a patient don't want to go to transplant because of, well, some problems, or because he or she doesn't want to go to transplant, I would say, okay, the overall survival is going to be the same. And the second important thing is the incorporation of the minimal residual disease in the future.
Excellent. So Herman, you are a transplanter, so maybe I can guess your answer, but who knows?
He's a CAR-T guy. He's no longer a transplanter.
No longer. No.
Sure.
He is a very loyal person. I know him now since more than 20 years.
So I agree with both of you. First of all, I think for most of the centers in Germany, transplant is going to stay. And you know that we are even doing tandem transplantation in high-risk patients, and this is still being performed. Also, according to the data that came from the EMN02 study. And I have only seen patients with really very long remissions, and I guess that they are cured if they received a transplant. And I always remember the Spanish study by Joaquin Martinez, who showed that patients that achieve a complete remission and remain in complete remission for quite some time, that they have a very low risk of relapsing. And I've patients actually in my practice that are out of transplant now for 10 to 23 years after transplant and still in complete remission. So I think at the moment, transplant for me is going to stay, but I'm with Thierry that all these novel immunotherapies are clearly challenging autologous stem cell transplantation. So I think the CARTITUDE-6 study, where we are going to compare upfront autologous stem cell transplantation versus CAR T-cell therapy, will be an extremely important study to show which way we are going to move. But I think at the moment, for me, transplant is going to stay.
So if tomorrow, for instance, Herman, the CARTITUDE-6 trial looking into auto versus CAR comes with a positive result, similar to what we have seen recently in lymphoma in favor of CAR T cells, do you think then auto would be downgraded into later lines maybe?
In certain countries, yes. But I'm not sure whether all countries will be able to afford CAR T-cell therapy first round. And so I guess even then, I think that would be a very sad development if the world is going to be divided into countries that can offer CAR T-cell therapy, which is more effective, and countries that can only continue to do autologous stem cell transplantation.
So Rakesh, in the UK, you are really very focused on cost effectiveness, and NICE has always a very stringent assessment of the different treatments. What are the current thoughts about the use of auto transplant versus all of these novel agents?
Yeah, thank you. That's a great question. And in fact, I was discussing this with some colleagues just yesterday, because as you quite rightly say, in the UK, the bar is on cost effectiveness and based on cost per quality or quality of life adjusted years. What we do know is that autologous stem cell transplantation is cheap. The facilities are in place. And you're going to have to see a very large magnitude of benefit, firstly, to demonstrate the effectiveness. The next major question is, what price will the CAR-T come at? And this is the bit that's going to need substantial negotiations to get the price down, to get the cost per quality in a reasonable place. Now, I must admit I am worried and concerned that in the UK we will not achieve that threshold and may not be able to access this therapy, particularly if you look at the way that the CARTITUDE-6 trial is set up, with DVRD and then DRD, thereafter. The cost of this treatment is going to be very problematic for the UK, and I'm concerned that we may not get access.
Excellent. Maybe I can direct a question to Dr. Bayliss, who has joined us. Good morning, Nizar. You're in Calgary. And we have a question here from the audience. So the debate is about auto or no auto, as you may guess. This is the Paul Richardson plenary presidential presentation at ASCO and late breaking abstract at EHA. And for all those guys who has just joined us, welcome to this post-EHA ASCO webinar focused on multiple myeloma by the IACH. So the question for you, Nizar, is from a biological standpoint, what do you think of this issue that we hear more and more about, the deleterious impact of melphalan, on generating novel alterations or more resistant clones? And then I'll ask the same question to Salomon, but looking into some of these myeloid diseases that were seen in the transplant arm.
Thank you, Mohammed, for the invitation. Good afternoon to you and to the rest of the panel. So this is the question. If we shouldn't do stem cell transplant, is it because it's going to be harmful? Let's say it's going to be equal to CAR T cell. Is there more harm of doing autologous stem cell transplant? And as you alluded, we do see from genomic studies a melphalan signature that many groups, especially Francesco Maura group, showed we can be identified in patients who received alkylating agents. Although there is no long-term data to suggest the signature has an impact on overall survival. If anything, most of the transplant studies show benefit in overall survival. And the argument there that when you use high-dose melphalan as a one-time dose, at that time the disease is being largely debulked, and therefore the cells are not being the myeloma cells not being exposed to high-dose alkylating agents. And this is in contrast when you use repeated melphalan dosing continuously or cyclophosphamide dosing continuously, perhaps. Having said that, though, if you look at the determination trial, you can see there's 10 patients with AML and MDS in the transplant arm versus no, 10 out of 330, 340. Is that sufficient to sway us away from transplant?
But I think if you look to the supplement, Nizar, there are also 7 cases of ALL in the non-transplant arm.
Correct. And that's biology there, because Revlimid is known to knock out Ikaros, and Ikaros can induce leukemia, ALL specifically in loss of Ikaros is one of the known mutations in ALL. So I don't think we have sufficient data to sway us away from transplant based on harmful effect in myeloma. But I would agree with my previous colleagues, esteemed colleagues, that the data from non-transplant, especially data from Thierry with the Maya. I mean, Maya gives you a median PFS close to 60 months. And now with determination with transplant and data VRD, we're getting also close to 67 months. Is 7-month difference in PFS worth it? Especially for patients who have good risk, standard myeloma risk? I would say probably no. So if a patient comes to me today who's 60, 61, say, can I just do DRD versus transplant? I won't feel too negative about this. And a high-risk patient, I think, is still more compelled to do transplant based on EMN2 trial, based even on data from the determination, especially for 414. 17P, we need to do what our English colleagues are doing with the optimum, perhaps an even more aggressive approach with better longer consolidation and so forth.
So maybe I'll change the order and jump to Thierry before bringing a question to Salomon. So Thierry, you are mainly in the elderly landscape, I would say, but I remember a few years ago, you attempted to show that even in the elderly, transplant may be useful, you know, and I remember the story of MEL 100, should we consider that that was long time ago, and this is now part of the history of medicine? And as mentioned,
I would not say the history of medicine. I could say the very small history of myeloma, maybe. But I know that this particular study had an impact in Germany because and Herman knows that, because transplant was not better than MP thalidomide, and it has a quite negative impact on insurance issues in Germany, as far as I know. No, but everything has been said to be honest, and I agree with what has been said. Just a couple of things. When you talk about transplant, you basically talk about melphalan 200, and your gain in PFS is 1 year. I know it's exactly what it is what has been observed in the vast majority of previous studies, in fact. So in the IFM part, we had 36, and we moved from 36 to something like 50, let's say 14 months. And here you move from 46 to 57 or 58, something. So basically your gain is 1 year. So you have, and the survival is the same. And then you have the CAR-T strategies coming probably soon. You have these. I agree that the discussion on hematologic SPM is not that clear, but you have 10 AML MDS versus 0 anyway. Then you have some more ALL in the VRD arm, other hematological malignancies as well. So the imbalance, if I remember correctly, the imbalance is 9 versus 13. But still, the 10 AML MDS are in the transplant arm. So you have the CAR-T, you have the SPM issue, you have the quality of life. So transplant is not is not a very nice experience for the majority of patients anyway. So year after year, you see some pushback for autologous stem cell transplantation. That said, it's of course it's still a standard of care for the younger patients, and I also agree that the CARTITUDE-6 study will not be enough. So we will need more studies. There is still a possibility that for some patients, you may be able to combine transplant on CAR-T strategies as well. And then one very important point is that transplant will remain, and maybe not that much in the US, but Melphalan 200 is extremely cost effective. What I would not like to see, and it has already been said, is that the poor countries will have Melphalan 200 and the rich countries will have CAR-T strategies or both. So that's but I am totally sure that transplant will remain on board for many years, I would say, for some countries, and possibly not that much for some rich and countries having access to everything or having access to the majority of strategies and drugs.
Okay, thank you very much, Thierry. Salomon, we have an interesting question here from James Kaufman about the determination trial, which showed similar overall survival and progression-free survival in those two cohorts who achieved MRD negativity, for instance. Can we use this criterion to decide whether to proceed to auto transplant?
Yeah, that's an interesting point. And of course, as you know, we'll have the MIDAS trial randomized exactly on this question that will help to answer this. What we can see from the data we have in the IFM 2009 and other trials is that perhaps a single point MRD might be not enough to have a very good prognostication of the outcome of the patients. And perhaps that, in the future, we'll use more and more the sustained MRD negativity as a, surrogate marker of PFS or overall survival, because it's probably a more powerful prognostic marker. So we'll see this, and we'll see indeed with, in the MIDAS trial, as opposed to determination or the IFM 2009, we improved the induction regimen because it's a quadruplet. We had the anti-CD38, so it can be different. And maybe we cannot transpose directly the data of determination to this new generation trial with quadruplets. And of course, we are looking at the non-transplant or transplant in patients who achieve MRD negativity rapidly after induction. And we know that the post-induction time points of MRD is more powerful. And we know that from Cassiopeia, right, than the post-induction, for example. So it's a strong surrogate marker. So we'll see in this context if it's enough to guide the treatment decision. We truly need these randomized trials to be able to answer completely.
Thank you, Salomon. And I'm glad you mentioned now that more and more we're doing quadruplet induction. And we have a question from Canada, from Dr. Neri, about the relevance actually of the current results of the determination trial in view of the fact that we are moving to quadruplets. Meral, what do you think?
Well, I think the quadruplets have certainly improved the response rates, depth of response. MRD negativities are very, very high. And although the French study and the Cassiopeia study have shown that frontline use has a major impact, even more than the post-transplant in the maintenance phase. So using DARA early, it looks like to be more efficacy, with higher efficacy, and also could be more cost effective. So quadruplets, so far, I mean, at ASCO, we have seen the updated results of Griffin. The MRD rates are remarkable. I think for future, we will be less considering about the cost because it will be a short term. And the cost benefit ratio will be very attractive, and it will be accepted by the authorities, health authorities, positively. That I think many will share my opinion. And its effects are visible for both high risk and also for standard risk.
Excellent.
So that's so far.
And I can add, you're right. For instance, in many places now, bortezomib is becoming a generic, lenalidomide is a generic. So we are, I mean, the main cost is driven now in the quadruplet by one agent, I mean, depending where you are. So before closing this transplant chapter, I would say maybe a question for you, Marie-V, and this is from Dr. Salinas, who says hello to all the panel and faculty, and he's very loyal to the ICH activity. Hello, Virgilio. So his question is about the Atlas presentation, and I didn't put the Atlas presentation as part of the key, I would say, findings. So let me reword it differently, because this is about the maintenance treatment. So Marie-V, where do you see the future of maintenance? Lenalidomide is the standard of care. Unfortunately, the Spanish trial randomizing ixazomib plus Landex against, compared to lenalidomide alone, didn't prove to be positive. However, we know from Forte that carfilzomib, lenalidomide, but also from the Atlas trial, presented at ASCO, that carfilzomib plus Len for 2-3 years, IV infusion, not ideal maybe. Where do you see the future of the maintenance treatment after transplant, assuming we will continue to do transplant?
Yeah, thank you. So I think the Atlas study evaluated the addition of carfilzomib to lenalidomide, I would say more as consolidation after autologous stem cell transplantation, with the greater possibility of stopping carfilzomib and dexamethasone in patients without high-risk cytogenetic abnormalities if they are in minimal residual disease negative after the first 8 cycles. So this possibility is quite attractive from my point of view, because what Dominic Ditfeld from Poland presented was how this approach increases the probability of achieving minimal residual disease negative. The progression-free survival was longer for KRD, and at the end of the day, those patients achieving MRD negative moved immediately to lenalidomide, and at the end, only high-risk patients or patients with MRD positive remain on maintenance with carfilzomib and lenalidomide. So I don't know how this could be translated into the clinical practice, because we know that this is something of label, but in principle, the idea is interesting. And at the end of the day, this clinical trial will meet with our final objective to have more and more patients in undetectable, measurable residual disease as part of the first line of therapy, if we consider that this is going to be an excellent prognostic factor predicting outcome in myeloma.
Meral, I see your hand raised.
Yeah, I have a question to Marie-V. Marie-V, what do you think if the induction was something more intensive than VDT, which is in the Atlas study? Would you still think the outcomes would be similar?
Maybe no, because it's true that in this study, the most common induction regimes were VTD or Cyborg-D. Well, maybe definitely we were discussing before, and if we move to bortezomib, lenalidomide, dexamethasone, anti-CD38 monoclonal antibody, or even carfilzomib in some patients, maybe this trial would not have any sense. But in countries in which the induction regimes will continue being these regimes, Cyborg-D or VTD, it would be possible to optimize the consolidation in order to put more patients in MRD negative, and well, without being necessary to incorporate the carfilzomib since the beginning, that we know that this is not approved in majority of the countries.
Herman, you would like to comment on this?
Yeah, I think we have two issues here. One is the patient's view, and I think what our experience is, and even German patients are not going to tolerate or take maintenance therapy for more than 2 or 3 years. So I guess maintenance until progression is a nice idea, but patients are not doing it. I think most of the patients, at least I know, they stopped maintenance after 2 or 3 years. And so this is the patient view. The other is the scientific view, and I think I'm completely with Marie. We should really aim for achieving MRD negativity, probably sustained MRD negativity, and then stop treatment. And I guess that a lot of trials that are ongoing at the moment are aiming for sustained MRD negativity. And I hope that the regulators will finally accept this as an endpoint, and this will help us to much more rapidly change and improve treatment strategies. And this will also be of great benefit for our patients.
Thierry, based on our lovely discussions we always had together, and I learned a lot from you, I could sense that you are very enthusiastic about bispecific antibodies. Do you see a role for bispecific antibodies in maintenance in multiple myeloma, and in which patient? Where would you love to see them?
This is okay. The first part is correct. I think all of us are somewhat enthusiastic about bispecific antibodies, but I don't really agree with the second part. I'm not quite sure the best place would be maintenance or prolonged therapy. I am still a little bit concerned by the fact that some patients will have got some severe infections, sometimes late infections, atypical infections, and I'm not sure. I'm not sure that patients will stay on bispecific antibodies for 2 years or even more. I think that will be these therapies will be extremely helpful for likely in the context of combination regimens, and likely for relatively short or not that long period of time. So that we don't know, but you know, if you manage a combined therapy for a very long time, including let's say some lenalidomide, a bispecific antibody, some one CD38, something like that, you will see adverse events. You may see infections, and you know, having a patient dying of infection, being MRD negative, being in sustained MRD negativity and dying from infection would be really bad news. And
we'll talk about the safety of bispecifics in a few minutes, but let me close the chapter about the maintenance by asking Nizar, because he did some nice lab work on all of this. What about, for instance, the new so-called cell modes, you know, the next generation IMiDs like iberdomide and mezigdomide? Where do you see a role for these agents in maintenance?
But biologically, Mohammed, as you know, they're clearly more effective from a affinity for cereblon or affinity for the substrate, Icaros and Iolos, and they're more potent immune inducer, especially for NK cell and T cell activation. So from biological perspective, yes, they appear better on paper, but we still need the randomized trial to see whether they're going to be more effective as a maintenance regimen. If I may go back to the bispecific antibody, I think I agree with Thierry that they're concerning about infectious risk, but it's an important question that we need to ask, because what is MRD? MRD is a disease that survived alkylating agents, survived immunomodulators, survived CD38 antibodies, persisted after treatment. And hence a novel approach, completely different, is important to test in this population. And I think one unique thing about bispecific antibody that Dr. Neri and our lab has shown, they not only induce direct killing of myeloma cells, but also they induce a proliferation of phenotypic T cell population. It's almost like you're vaccinating against a tumor because you're expanding potentially a reactive T cell clone against the tumor. So perhaps a limited duration of bispecific antibody until MRD, sustained MRD is achieved and you stop would be an important question to ask. Safety is paramount. I agree with Thierry. We need to ensure they are safe. But I think it would be important to test in this MRD positive population, perhaps along with novel cell mods as well.
Okay, so let's move now, guys, to the treatment of high-risk multiple myeloma. And what has been really amazing for me, and I thought it's really a good move to see over the last couple of years some dedicated trials to high-risk multiple myeloma, and we'll discuss maybe how to define high-risk multiple myeloma, although everybody agrees that plasma cell leukemia is high-risk multiple myeloma. When it comes to cytogenetics, things are a little bit more complicated. And we know, for instance, the Optimum UK9 trial by Dr. Kaiser and colleagues has been really nicely showing some good results, and it's a very heavy package of treatment. The German concept trial is also another good example of that you can intensify the treatment for high-risk multiple myeloma. And last but not least, we've seen at ASCO and EHA the IFM 2018-04 trial by Dr. Tuzo, who could not join us tonight. He had computer issue, I think. So Rakesh, what is the current practice in the UK? Are you still including patient in the Optimum, or you switch to maybe a more gentle approach like in the concept or IFM trial, or are you still hammering them as you did in the Optimum trial?
Okay, so the Optimum trial has now closed, and that recruited very rapidly, and we're now facing the aftermath of what happens when these patients relapse, which is another section to talk about perhaps later. But so I think there's two ways to look at this. The first is, what are we doing on a routine basis? And the second thing is, what are we doing in the context of a clinical trial? So on a routine basis, unfortunately, we remain slightly limited. We have induction with DVTD, and then what we would do is a tandem transplant based on the EMN02 data. And then currently we're following that up with Revlimid maintenance. There is some of us have been moving straight onto KRD, a little bit similar to the Atlas approach following the tandem transplant. So that's kind of the standard approach, with the idea of trying to maintain a triplet sort of maintenance consolidation approach following a tandem transplant. Within the context of a clinical trial, we have the RADAR trial, which is currently open, where patients are genetically stratified. We have a different way of stratifying patients on this. So in Optimum, patients underwent Sky92 profiling, and those with two high-risk features of Sky92 high risk were classified were allowed entry, whereas in RADAR we're using the standard approach, the IMWG approach, with the addition of chromosome 1 abnormalities. And those patients will, in the amendment, get isatuximab with CVRD as induction, followed by a transplant, and then go on and have isatuximab Revlimid maintenance. So that's kind of the approach that we're following. It's similar to the Optimum approach, with this kind of dose-dense, intense, prolonged treatment approach.
So in the Optimum, it was DARA, VRD, and cyclophosphamide.
Oral cyclo, yeah.
And in the concept trial, Herman, it was isatuximab KRD. And in the French trial, it was daratumumab KRD. So Salomon, where do you think, I mean, obviously, maybe you're biased. You want to say, well, the French approach is the best one, but I'm not 100% convinced that actually these approaches are very different, by the way. What do you think, Salomon?
No, it's an interesting question. And, you know, of course, I think we will never have comparative trials, so difficult to say which one is the best. But it was interesting to see the data from the concept trial last year and the IFM 2018-04 trial this year with Cyril Touzeau, which are, you know, quite similar. So basically we had the response rates and CR rates after the induction, 6 cycles of DARA KRD or ESA KRD, and we reached, you know, very high response rate. It was almost 100%, and almost all the patients achieving at least a VGPR. And we had this MRD post-induction MRD status at 60%. So which is of course very good for those patients. So of course the follow-up you know, is still lacking in those trials. So we need to see And of course, we suspect that those high-risk patients, they can respond well initially, but relapse, rapidly during the course of the treatment. So the real challenge is to maintain their response across the time. We tend to do as well tandem autograft stem cell transplantation for those patients. And we tend to keep lenalidomide as well as maintenance. Sometimes, people, as Rakesh just said, sometimes people try to do more in maintenance. It's also after tandem transplantation and strong induction, it can be sometimes difficult, in terms of cytopenias, to intensify too much the maintenance, and if we want to keep it for long term. So we use lenalidomide as, it's the label for maintenance in France anyway. So it's surely an interesting fact that we start to have more and more data on these very challenging patients.
But, my remark about the subtle difference between concept and the IFM 2018-04 daratumumab KRD versus isatuximab KRD was mainly related that in the IKEMA trial, there was a sub-analysis showing that isatuximab may be a good drug for 1Q amplification. So let me ask Marie-V from outside, because otherwise, these guys that would advocate for their own trials. What do you think? Does, for instance, using isatuximab can bring maybe more added value, having these data in mind about the 1Q amplification?
Well, honestly, I think that it is difficult to answer to this question and to say yes, because definitely this is something that has been evaluated specifically in the clinical trials with isatuximab, and maybe this should be also evaluated in the clinical trials with the daratumumab, definitely to distinguish plus 1Q from amplification of 1Q. And the other thing is the association of amplification of 1Q with other high-risk cytogenetic abnormalities. And I would say that concerning the high-risk myeloma, I completely agree with you. The good news is we are observing more and more specific clinical trials conducted for this population. And I think that I like the Optimum trial, as well as the French study, the concept study, but I think that we have to do a next step and to incorporate the immunotherapy and to expose to the plasma cells to different drugs with different mechanism of action, incorporating maybe the BCMA targeted therapy and so on, especially in those patients in which the residual disease is present. But this is, of course, this is something that we'll see in the future.
Okay, so we have one comment from Miral, and then I'll come back to you, Herman.
So my question was regarding to the panelists about how you approach for patients with single hit or double hit in terms of high risk. So high risk is certainly not homogeneous. And the other point I would like to remind that in the Fortis study, 1Q abnormality did not benefit from carfilzomib either. So the weight in the DARA KRD or ISA KRD relies on the anti-CD38 component. So these are the two issues I wanted to draw attention to.
So maybe Herman, you would like to comment on this?
First of all, I just wanted to really explain that the concept trial is still ongoing, so we are still recruiting. And in addition, we finished the recruitment in a study with VRD plus ELO and another study KRD plus ELO. So we can now really compare in high-risk patients the role of Velcade versus carfilzomib, and also the role of the addition of an anti-CD38 antibody in high-risk patients. So maybe we get some information about the role of certain drugs in high-risk patients.
Any role for CAR T cells in high-risk multiple myeloma? Thierry, you're smiling, I'm sure.
No, I was thinking, Rakesh started to say that we do what we can, and looking, for example, if you look at the MIDAS study, you could say the best part is possibly not the part for high risk, because for high risk patient, the randomization is single versus double transplant, which is, you could say this is possibly not optimal. The only thing is that the maintenance has been optimized, and the maintenance is iberdomide with isatuximab, let's say. So, okay, we do what we can do. I agree with you, and I agree with Marie-V that we have now some kind of we have an opportunity, in the sense we have different targets. And looking at high-risk patients, I do believe they need some kind of prolonged therapy or maintenance therapy for a quite long time. But they probably, that would be a good idea to play with different targets. So we don't know exactly how to do that, but switching the target, having a BCMA for a while, then switching to GPRC5D or FCHR5, or why not PD1 or something like that, possibly implementing CAR-T strategies as well. So we have tools to be innovative for high-risk patients, and this is something we did not have at the time we designed the MIDAS study. We did not have access to that in a way.
Excellent. One last question to you, Nizar, because we need also to have some, I would say, real world information. If tomorrow morning in Calgary, you have a 60-year-old patient who, you know, present with deletion 17p multiple myeloma, fit, no comorbidities, and okay, we'll close the debate about the percentage of deletion 17p's. This is 75%. What would be outside the clinical trial, the treatment you are offering to this patient at your institution?
The induction is still the same. We still only have VRD funding, followed by transplant. One or two will discuss with the patient about 1 or 2 transplant. I wouldn't push too much for 2 transplant, as long as I can use continuous consolidation VRD for at least a 2.5 or 3 years consolidation, as per the total therapy 3, if you want, or the data from the Emory group, because that's what's funded. It's not optimal, obviously, but that's what's funded. But I clearly believe immunotherapy is the way to go for this high-risk population, especially the 17p deletion. And we see from the CARTITUDE trial that the majority of the patient has sustained and deep remission. Now, the high-risk population definition may change with immune therapy. We need to understand who are these patients failing eventually. But I think immunotherapy is the way to go for this high-risk population.
Excellent. Thank you very much. And actually, that allows me to have a very nice transition, actually, to the immune therapy big chapter, which is going to be our next part here. And this is going to be about bispecific antibodies and CAR T cells. And I'll start with bispecific antibodies. So we have the big group of anti-BCMA bispecific antibody T-cell engagers, but now we see more and more of the anti-GPCR5D T-cell engagers. And for instance, when it comes to the anti-BCMA T-cell engagers, we heard some very nice data about teclistamab, but also teclistamab in the the global cohort, which hopefully will lead to the approval of this bispecific as soon as possible, at least. I mean, this is my wish. But we have also subgroup analysis in those patients who were pre-treated with an anti-BCMA directed agent, which I believe is a crucial piece of information. We have also actually very, very interesting data, including nice safety about elranatamab. And that was the presentation of your presentation, I think, Nizar, at ASCO. Of course, there are many others. There are also the Regeneron one, which results are very interesting, especially in terms of safety, because the cumulative incidence, for instance, of CRS of the Regeneron agent is almost half what you see in the other agent. I don't know if it is just by chance or if it is true. So regarding the anti-BCMA agent, Herman, what do you think? any favorite topic, any favorite agent, or where do you use them today, and how do you see the future of these agents?
Now, I completely agree with you that the T-cell engaging strategies, whether they are CAR T-cells or bispecific, are really going to change the treatment strategy for our myeloma patients. Now, if you look at these different BCMA targeting bispecifics, clearly they seem to be about the same, slight differences in overall response rate, slight differences in the rate of complete remissions, slight differences in grade 3 CRS or neurotoxicity. So at the moment, I don't see one of these bispecifics to be really more effective or safer than the other one. So I think the race will be which one is going to be the first one to be approved. And here I clearly see teclistamab in the lead. And we'll have to see whether the other companies, by maybe very good strategies in further developing their bispecifics, will make it. And then it will be probably the price that is going to decide about which one is going to be used. I think, which is what I think is very important, is that we have new targets now, and you alluded to the GPRC5D, you alluded to the FcRH5, and I think we're going to see more and more target antigen loss. We have now already 5 patients that have an irreversible BCMA loss following CAR T cells and bispecifics, and Nisa has and his group, they have nicely shown that there are different mechanisms of resistance to bispecific antibodies. So I think we are going to see immune escape, and we need other targeting structures that we can also target with bispecific antibodies. So I think it's an interesting race. I think BCMA is probably soon clear which bispecific is going to make the race. We probably still have to consider the BMS bispecific which has 2 binding domains. So maybe if we look at the CAR T cell field, maybe the dual targeting will make a difference. But I think the other one is the race between the others is really who makes the first one to be approved, and the other targets. I think it's very important that we have additional targets.
So you mentioned Herman, your experience with the loss of BCMA. Here I'd like to ask Mary V, what's her take on the analysis of cohort C from the teclistamab, the Majestic trial, which actually could show that if you have been exposed to belantamab, the antibody drug conjugate against BCMA, or even to CAR T cells against BCMA, you can still achieve 52 to 55% overall response rate. This is slightly 10% less than what you see in the general cohort. What do you think of these results? Because that's going to be important, I think, for the sequence of the different agents. We know that now belantamab is increasingly being used because there is a, I would say, learning curve, and we know how to better manage its ocular, the keratopathy, by adjusting the dose, by increasing, I think, the timing. So belantamab, so patient will be exposed to belantamab, and hopefully some patient would have received also CAR T cells because they love the one-shot treatment and the treatment-free interval. So what do you think about these results, teclistamab after exposure to an anti-BCMA agent?
Yeah, so I think that this is a proof of concept indicating, as you mentioned, that the teclistamab was effective in patients previously exposed with BCMA targeted therapy, and not only antibody drug conjugated, but also 15 patients had been previously exposed to CAR-Ts. And the overall response rate is true that is a bit inferior in comparison with the teclistamab in the MajesTEC-1, but it's quite good, and over 50% across the different subgroup of patients. And I think that this is what we've seen. Of course, I would say that in the future, it would be ideal to evaluate very well the mechanism of resistance, for example, to CAR-Ts, in order to plan the optimal sequencing. And in addition, we have also some interesting data combining the BCMA CD3 bispecific monoclonal antibodies or GPRC5D with other drugs like daratumumab. So maybe in the future, this would be even more attractive than to rescue these patients with a teclistamab single agent. And again, I see quite logical to rescue patients previously exposed to belantamab mafodotin with teclistamab, assuming that the BCMA does continue being present on the surface of the plasma cells because the mechanism of action is different. Concerning the CAR-T followed by a bispecific monoclonal antibody, we've seen in this cohort how this is feasible, but I would like also to incorporate the T-cell exhaustion, because if this would be the mechanism of resistance to the BCMA CAR-T, well, maybe this would be better to give something in between in order to restore the immune system and to consider the BCMA CD3 for the subsequent line of therapy. But this is the theory, and in principle, in this study, the sample size is, not very high, 40 patients, but I think that it does demonstrate that it is possible.
Excellent. So Rakesh, in the UK, you have really, I was very impressed with your experience with belantamab in salvaging these patients. So what is your current view on the outcome of the patient who received belantamab and then progressed? How do they respond to other treatments in general, in your hands?
Yeah, it's a great question, and we've started to get these sorts of patients who have moved on. In general, the results that we're personally seeing, and this is anecdotes, so just need to remember this, has been positive. So we are effectively salvaging patients who've had previous belantamab with either bispecifics or indeed CAR-T. So I think doing it that way round, given that the mechanisms of action are so different, it does seem to work. What we haven't done, and what we don't routinely do, is test for BCMA expression, because that's always the question that comes into mind, is that if you have a patient progressing on belantamab, do you need to test that they are antigen positive before you move to another BCMA agent? And we haven't done that. And we haven't seen particularly a problem. So I think you can do that, and certainly we're also salvaging patients with GPRC5D T-cell engagers post belantamab, as you'd expect. One of the things I am just a little bit concerned though is about infection. So if you've got two BCMA agents back to back, what sort of infection risk are you getting? Because there is hypergammaglobulinemia with belantamab. The infection risk doesn't seem to be as high as the BCMA bispecifics, but surely there must be a cumulative incidence of infections when you do this.
Salomon, you looked to me earlier in this webinar to be a little bit more optimistic about this issue of opportunistic infections, because obviously I have seen these patients who are receiving the bispecific antibodies are really very advanced. They have received tons of dexamethasone, highly immunosuppressed. So for me, it's very shortcut. It's a shortcut to say, well, if they developed an invasive aspergillosis, suddenly this is, they have been in a neutropenia for several weeks or months. So what are your thoughts, Salomon?
No, absolutely. I mean, I think we definitely need to be very careful with this risk of infection. We are on a learning curve, and we need, of course, to accumulate more and more data. But as you mentioned, those trials on bispecific were done in patients who had received several lines of treatment. they can have a T-cell dysfunction due to the previous treatment that they received, and in the opportunistic infection that we see, like CMV reactivation, PGAP, for example, it's often linked to T-cell dysfunction more than hypogammaglobulinemia. However, we can think that the B-cell and T-cell interaction is perhaps missing when you treat patients with BCMA targeting therapies, and that can also play a role. So it's difficult. I think we need to accumulate more and more information. The main message is that we absolutely need to be careful, and on the ongoing trials, I think we need to have very careful prevention of infection. So we know that patients who have deep hypogammaglobulinemia are more at risk of infection, so supplementation with IVIG is always a strong recommendation. And perhaps in some patients, it would be good to follow the CMV in case of positive serology, for example, or in case of symptoms, to make sure that there is no reactivation. My hope is that again, it will be less of a problem at earlier lines of treatment, 1, 2, that we will learn enough to manage this well, to know when we should supplement with IVIG, to know which patient we need to follow for different opportunistic infection. And we have the treatments for CMV, so we can treat the patients if we see CMV reactivation. So I hope that we will be able to manage this, and it won't be too much of a problem for those treatments that are, of course, very useful in those patients.
So Nizar, these bispecific antibodies are moving very quickly into earlier lines of therapies, and even they are now being combined. And we've seen, for instance, the TRIM trial with actually anti-CD38 monoclonal antibody. So given all the words of caution we are hearing about prophylaxis, etc., do you see an immunological rationale to be worried that by going into frontline and by combining them with anti-CD38 monoclonal antibodies, we should be even more worried? And then I will ask Meral what would be her ideal prevention, regimen, whether antifungals, antibiotics, IG supplementation. What do you think, Nizar, about the rationale, the immunological basis for this?
Well, the rationale for combination is clearly there, especially with anti-CD38 antibody and immunomodulators. The rationale for anti-CD38 comes from work from Niels van de Donk and some work from our German colleagues as well, where they showed that CD38 potentially targeting Tregs, CD38 positive Tregs, but also some Bregs. So that could be the rationale. And obviously the addition of immunomodulators activating T cells and NK cells. We know from some CAR T cell data now that NK cell even are important in the CAR T cell context to enhance the immune response. So I think biologically the rationale for combining them. Does the increased risk of infection? I would say definitely adding daratumumab going to knock out your NK cells as well. So there is potential concern about worsening infection in this patient population. I agree with completely what Solomon said. We're seeing quite a bit of viremia, but what I'm struggling to know, are these viremia causing disease or are just transient viremia resulting from redirecting T cell therapy? I think I said this example in many venues. I had a patient who showed up on a Wednesday feeling unwell, tested positive for CMV viremia, over 100,000 copy by PCR, supposed to pick up his valganciclovir, didn't pick it up until the following Monday, came back the following Monday. How are you feeling? I'm feeling great. Didn't pick up the valganciclovir, repeated the PCR, viremia completely disappeared. So I'm wondering if we are all being overcautious in some of these patients. We're seeing quite a bit of EBV, CMV, parvovirus, especially parvovirus in this population, whether it's all niche therapy or withholding therapy or simply observation. We need to learn more how to manage that. I'll just add one more quick thing about the BCMA loss. I would be cautious about minimizing that impact though, Mohammed. With the work we're doing, we're seeing quite a bit of not necessarily biallelic loss of BCMA, but now we've seen 2 cases of single amino acid or single codon mutation in the extracellular domain of BCMA. And one of them is arginine 27 to proline is quite disabling the activity of all the bispecific antibody we've tested. So we have to be careful about in the future not simply looking at the BCMA expression by immunohistochemistry, which stains the intracellular domain of BCMA, but looking potentially at mutation in the extracellular domain, which can also affect the activity of these bispecific antibodies.
No, I fully agree. And actually we are learning, I think, while we are I would say. It's like learning to swim while swimming. So Miral, what are your thoughts? What would be your advice to the colleagues when it comes to supportive care in those patients who are at very high risk of opportunistic infections? I know, I mean, our community is able to do bispecific CAR T-cell trials, randomized trials, but we were not able yet to settle the issue of antibiotic prophylaxis. I know Rakesh will disagree and tell me the UK colleagues did everything and, you know, and they found the answer, but I'm not convinced 100% yet, especially that things can be different in different environments. What is your advice, Miral?
Well, I think Nisa already answered most of the issues. I mean, the viral reactivation is certainly of importance. And during the COMI meeting, he shared his thoughts about the parvovirus, about when approaching a patient with, unexplained cytopenia. Other than CMV, parvovirus can be an issue. But I'll come to another point. I mean, we have been experienced in allogeneic transplant for so many years. And we are so much accustomed to handling immunocompromised patients. And I think this is an era that we have to revisit our old experience, but of course with new microorganisms. I don't know how much value does continuous antibiotics have, but Niels in another meeting, he shared his thoughts, and also during the EHA meeting, that he prefers to give continuously Bactrim. For us, we use it 3 times a week. So this is another approach. It could be, from an experienced person's perspective. So I think in each center, people should develop their own modalities to overcome. We are using IV immunoglobulin very frequently, and we have participated. We are still in the DREAM8 study. That's a problem there, and we are now starting with the bites. I'm sure we will share the same experience with all the other centers are facing with neutropenia, with lymphocytopenia, and with hypogammaglobulinemia.
No, I agree with you. And to be honest, I must confess that in my center, for instance, we made things very simple. We just applied, the allotransplant SOPs to be on the safe side, and things are rather good in my opinion, but time will tell. I have a difficult question for you, Marie-V, still regarding what we're talking about, and this is a question from Dr. Ed Cliff. It's about the methodology. So in myeloma, we are highly familiar with progression-free survival as an endpoint, but now we're talking about opportunistic infections maybe, and maybe other side effects. And I guess, although Dr. Cliff didn't mention this, the idea is, shouldn't we move to event-free survival endpoints? What are your thoughts? I mean, you've been running dozens of clinical trials in the last 15 years or so. What are your thoughts about this progression-free versus event-free? We debated overall survival versus PFS earlier today. What about now incorporating other events?
Well, we moved from a minimal residual disease, and we are fighting in order the authorities to recognize MRD as endpoint, but we can move to event-free survival, including the infection. So I don't think so, but definitely we have to take this information into consideration. And from my point of view, these infection rates are between brackets acceptable in the population in which these clinical trials are being conducted. But definitely when we move to earlier lines of therapy, maybe, I don't know what the rest of the panel will consider, but we have to go to fixed duration. And as soon as we are observing excellent responses, many patients in minimal residual disease negative, maybe we have to plan stopping rules when the patient achieves minimal residual disease negative, or what we consider optimal. And indeed, we are observing, and we have in the clinic some patients that they had to stop the treatment with bispecific monoclonal antibodies because infections, and especially COVID-19. And yesterday I've seen a patient that has been since Christmas more than 5 months without teclistamab and talquetamab together because of a persistent COVID-19 positive with other infections, and surprisingly, the patient does continue in complete response. So I think that this opens the door to plan a fixed duration of therapy, and this can potentially help to improve the safety profile.
So just before closing the debate on the anti-BCMA bispecific and move to other targets, Salomon, what are your thoughts on the elranatamab data? I mean, I was quite impressed with these results, because although we compare, you know, the anti-BCMA agent, nevertheless, we should bear in mind that in the Majestic trial, the median of prior lines of therapy was 5, whereas in the elranatamab trial, they were more advanced. It was 6 and even beyond, and some of them received CAR T cells, which means they were harder to treat, I would say.
No, I agree, and you can say that there is more a triple class refractory patients, but you know, at the end, it's still like, you know, small number of patients, or you know, early phase of the you know, trial developments. So it's difficult to compare. You know, my general impression is that they look to have a similar efficacy. We see 63 overall response rate in one side, 60 in the other side. They didn't give us much details actually on the response rate in terms of depth of response. And elranatamab, I mean, the Magnetism 3 was with a very short median of follow-up time. You know, for this report, it was 3-point-something months. So we don't know, we don't have information yet on survival and how the, you know, those 3 response maintain across the time. But we'll see later. But they seem quite similar to me, all of them.
Excellent, thank you very much, Herman. Let's move to the GPR C5d target. Of course, we already know the data about talquetamab, and talquetamab is even now being combined, as I mentioned. But at the last EHA meeting, I was quite amazed with a new player, I would say, a newcomer, new kid on the block, the Regeneron 6234, and that was a first in a human trial in 51 patients, which is already quite a decent number. So what is your take on these different agents targeting GPRC5D? And do we have any clue about now sort how to sequence them? You know, if you have an anti-BCMA available and an anti-GPRC5D, do you think you should start with this one first or with another one, or it's just by chance?
It's a very tough question. I don't think we really have the data yet. So what we know is that the efficacy seems to be quite similar. The toxicity is definitely different. From the data that we have seen, but I think we haven't really seen a lot, it looks like BCMA targeting is affected with a slightly higher infection rate. On the other hand, with the GPRC5D, you clearly have additional toxicity, like the skin toxicity, nail toxicity, mucositis, dysgeusia. And we had several patients that receiving GPRC5D bispecifics actually had to stop treatment for these skin and nail and also membrane toxicities. So this is a different toxicity that we don't see with BCMA targeting. I think we really have to compare whether this infectious problem is clearly in favor of GPRC5D. That would be an advantage, but the skin nail toxicity, the mucous membrane toxicity is an issue with GPRC5D. So at the moment, we tend to use BCMA targeting first and then switch to GPRC5D. But on the other hand, you take what you have. Sometimes the studies with GPRC5D are open, so we include patients there. and the BCMA is not available. But I think the story is not closed yet. But I think that the additional toxicity of GPRC5D is an issue. And then we haven't actually used the FcRH5 targeting. But Lisa, yeah, I think you have a lot of experience with this bispecific. And do you see additional toxicity apart from infections and CRS and neurotoxicity with this agent?
So this is about cevostamab.
Yeah.
Yeah. No, Herman, we don't see any additional toxicity like we've seen with GPRC5D, because as you mentioned, the expression of GPRC5D on keratin tissue, it's mainly cytopenia and infection that we've seen with cevostamab. Slightly more, as you know, which caused the latest progression, I guess, development, slightly more CRS initially, but now with the better step-up dosing and the use of dexamethasone, this has been largely mitigated. Interestingly, I mean, GPRC5D conceptually may have one advantage. BCMA being shed in the serum may cause chronic T-cell stimulation in the context of the bispecific antibody, maybe lead to potential more exhaustion, while GPRC5D not being shed. Is this potential conceptual advantage? I don't know. But like Herman, we've used larger GPRC5D after BCMA bispecific because of the clinical trial availability, and we've rescued large number of patients post-BCMA failure with GPRC5D. We haven't done the reverse yet, Mohammed, so it'd be interesting if you alternate or use GPRC5D first or not. And again, whether this BCMA shedding has any impact on the efficacy of these bispecific antibody and T-cell exhaustion would be interesting to see.
I like the previous comment from Herman, because this is important, and there are many patients actually attending this webinar, is that on one hand, you may have life-threatening infections, but on the other hand, we should not neglect not life-threatening infections, but things that can really alter the quality of life of the patient. Because I think all of us, unfortunately, who got the COVID-19 infection and lost, you know, the taste, I can tell you, it can be a nightmare for a few weeks if you keep on adding salt and you can't feel, you know, but that's another story. But I think this is really important, because we're going to treat these patients for several weeks or years. So that brings me, guys, to a sort of last 5 minutes to wrap it. And I would like to concentrate on the CAR T cell versus bispecific match. Of course, I know it's not a sport match, because our patients are really very brave, and they are really, you know, taking all of these aggressive treatments with lots of resilience. And I have really huge respect to them. But when we look to this, I would say, match between CAR T cells bispecific, when it comes to the efficacy, they look to me both very good. Safety, I would say, okay, let's say manageable. Cost, well, it has been always very high, and everything is increasing these days. You know, inflation is everywhere. Time to initiate treatment, CAR T cells, it can take you from 1 to 4 weeks, even with a speedy CAR or fast CAR. When it comes to bispecific, well, within 1 day, few hours, a couple of days. You need bridging usually with CAR T cells. It's not needed with bispecific. Of course, you need lymphodepletion with CAR T cells, especially fludarabine, kidney insufficiency. No lymphodepletion needed with bispecific. Convenience, you know, single shot, one and done, but you have to be in hospital for some time. For the bispecific, maybe short hospitalization, but probably longer treatment of duration. Also, I appreciate Marie-V mentioned that you can keep MRD negativity after bispecific without any treatment. So what are the thoughts of the panel about these two, I would say, families and players, and how are you going to use them in your practice if they are available tomorrow? Let's start with you, Marat.
I think, I mean, of course, it would be an ideal word to discuss one versus the other, but currently there is a shortage of vector, and CAR T is not available in every country. So taking this into account, if there is no problems with the bridging part, and I mean, you have it in your hands both at the same time, in that case, I think CAR-T would be the preferable one, because you give a treatment and then you can be safe for 18 months or so without any further treatment, with only follow-up and monitorization. I think every patient would prefer this.
Rakesh?
Yeah, I agree. I mean, the headline on some of the CAR T-cell data is extremely good, with very high response rates. And with cilta-cel, we're still awaiting the progression-free survival after 2 years still. So you can't really argue with that. And from a patient perspective, someone messaged me quite recently and said, well, could you get cilta-cel and then you relapse after 2 years and get another dose of it? Because it's clearly very attractive to be able to do that and keep yourself going for 4 years if the science works behind that. It may not. But, and you compare to the bispecific, where the patient is continuously entering, then you can see these huge advantages. But again, as Morel says, it's not straightforward. How do we manage those aggressive relapses? How do we bridge them, which I'm still really struggling how to bridge these patients. And of course, you have these elderly patients, the frail patients, and the renal impairment patients who are not going to go to CAR-T. So I think it's a great option, but it's for the very select few.
Mary V, what is the Spanish perspective?
Well, I think that the Spanish perspective will be quite similar to all to the rest of the centers worldwide. I would say that.
So we are all in a big mess, you mean?
No, I think that if the patient is eligible for CAR-T, from my point of view, this would be the first choice, especially because of what we see right now in clinical trial is how patients remain disease-free and treatment-free basically in the first month after the CAR-T administration, and this is impacting in the quality of life. But of course, we have to realize that not all patients are going to be eligible for CAR-T, and maybe more aggressive disease that they can't wait the manufacturing process. Older patients with some comorbidities, definitely they can receive the bispecific monoclonal antibodies. And at the same time, it's true that the potential lower efficacy with bispecific monoclonal antibodies can be compensated by the possibility of being combined. So honestly, I don't know. I think that we have to put into a balance the patient and the disease-based factors, as well as the familiar support where the patient is living, many factors. But the good news is to have all these approaches is available, and we will select them.
Excellent. Herman?
Yeah, I think a lot of the points have already been addressed, but I think the patient's perspective to have and I'm with Marie V, the one-shot therapy and then being treatment-free for a very long time, that's very attractive with the CAR-T cells. And
but can I be very provocative, Herman? Do you believe that in the future we will continue to deliver CAR-T cells and then do nothing after it? Don't you think that the maintenance concept will come, you know, someone will bring it after CAR-T cells? And I think it's already there. Come on.
Yeah,
you are right, but I think then the beauty of the CAR-T cells will really be clearly decreased. I think if we need additional treatment after CAR-T cells, then the question really comes up: what's the advantage versus bispecifics? But I think we need more data, and I think also the CAR-T cell technology is improving all the time. So, and maybe we can also re-infuse CAR-T cells maybe after 2 or 3 year time. So that could be also another opportunity. So I think at the moment it's difficult to say, but still the beauty of a one-shot therapy and long-term disease-free survival, that is clearly in favor of the CAR-T cells.
And that's true. That's true. This is really has been witnessed by many patients, and they feel like they are free of disease for some time. Nizar, any thoughts on this?
No, I largely agree with what my colleague said. Given today, if a patient showed up in front of me, I have choices between a cilta-cel or a CAR T cell versus bispecific antibody, frailty on the side, everything on the side. I think CAR T is the way to go. Depth of MRD and so far durability of response is clearly superior with the data. We need longer follow-up, obviously, but CAR T for now is the winner. As Herman said, the field is in infancy, so we're going to have better CAR T cells, especially improved persistence, will hopefully negate the need for maintenance therapy post-CAR T for improved persistence of these CAR T cells.
Excellent. So I left deliberately the last word to Salomon because he is very modest and humble, but he is our newly appointed professor in France, and I'd like to congratulate him. Very young, very brilliant guy. So Salomon, what's your view?
A lot of pressure here. I mean,
but you are the future, you know.
Lots has been said, but no, just in general, I'm very impressed by the CAR T cell data, and we've mentioned the CILTA cell data that are truly impressive. You know, in fact, in real life today, in the relapse refractory setting, what we can say from countries that have access to commercial CAR T cells, it remains a challenge, to treat patients with CAR T cells because we still have limited access, because all the reasons that has been mentioned of rapid progressive disease and etc. So I think it would be very good to have also bispecific antibodies, and they would be used, I'm sure, quite extensively as well in this context of relapse refractory setting, and that would be very useful for our patients.
Well, excellent. This has been really a great discussion, guys. We can spend another few hours actually debating all of these fabulous data. You all did a great job, and I'm very, very grateful for your critical and deep thoughts on these data. I mean, the progress in multiple myeloma is really amazing, and I think this is about the United Nation against multiple myeloma, as Professor Anderson always used to say. So thank you very much. Thanks to our audience. I hope you have enjoyed it. I learned a lot from these discussions, which have really a very nice flavor. So thanks a lot to our panelists who really did a great job, and I hope you will continue to support the IACH activities, and you will join us for some future events. I think next week we will have a very nice journal club, I think, on a JCO paper by Bruno Peva about circulating tumor cells in myeloma, but we'll have also another webinar also next week, I think sometime on Thursday, with Niels van de Donk about some of these immune therapies, where we'll dig a little bit deeper into the data. In the meantime, I wish you all a nice evening, a nice day, a nice afternoon, wherever you are. Please stay safe and keep well. Take care.